Elmer Fernandez from the Universidad de Córdoba in Argentina, and he has a very strange title, Who is There? The New Paradigm of Molecular Signatures. Thank you, Elmer.
Thank you very much for the invitation. I'm very happy to be here. Montevideo is a city that I love. And also it's very nice to meet people that I usually collaborate from Chile, Uruguay, Brazil. So I'm very happy. So and also I would like to incorporate more collaborations with all of you and with Europe in the future for all of us. So my name is Elmer Fernandez. I'm leading the Bioscience Data Mining Group in the Catholic University of Córdoba. I belong to the CONICET, which is an institution that hires research scientists across the country. And this is the institute that I'm working in, is a center for research in infections, immunology and infection disease, which is the leader of this institute. We are working together in the development of vaccines based on virus-like particles. So I'm not going to talk about, well, my group is focused on the joint venture between statistics, machine learning, and computational skills. My primary research line is focused on the integration of multi-omics data sets and multi-cohorts. So in this sense, I work a little bit in the same line as Dr. Nakaya. We also develop tools to integrate from a functional point of view instead of looking for specific genes, which is called MIXA, that I would like to found someone who wants to develop the interface because it's basically for bioinformatics. So we want to develop this user interface in the next future. But I'm going to focus in one of the things that we are working right now, which is trying to know a little bit more about the immune system in tumor samples. So from the beginning of this century, molecular signatures was thought as a kind of the trend line for better diagnosis. So hundreds of molecular signatures were developed in this period. Around 95 or 99% of them failed. And at the beginning, the molecular signature was defined as a set of biomolecular features, genes, mutations, DNA sequence, etc., which of course should be attached to a computational procedure. In those days, the main target was to predict a phenotype of clinical interest. So the main objective was to identify or to see how this patient's going to behave in the future. So if he's going to have a relapse or no, or if he's going to survive a long time or no. So, for instance, in breast cancer, a little bit of success on these molecular signatures, which are already in the market after around 15 years of work from the first appearance in the journals. So that means it's a lot of work for translational medicine. And the main objective of this molecular signature were basically to know who you are. So if you are in this stage of the disease, and if you are going to have a long survival or not. But thanks to these guys, we realized that tumors are more than tumor cells. So a tumor is a bunch of friends. So it's a community. And the tumor cells integrate in this community, and of course, they chat and negotiate with this environment. And they talk with the guy who brings the food, they talk with the guy who brings the oxygen, and who build the ways, the routes. And also, more importantly, thanks to this guy, we realized that they also talk with the police. If you want to do a crime in a specific area, you need to clean up from the police this area. So they should negotiate with this environment. So at the end of the 70th decade, Roger Waters was felt enclosed in a wall, and they asked, is there anybody out there? But now we are interested in who is in there. So we are now asking who is in there, these walls, and who are they? Why? Because if we know who are they, we can figure it out, how to talk with them and to convince them to help us. In the tumor, we can do this by means of, for instance, flow cytometry. But to do this, we need the sample, which is inexpensive, in order to make it in a big scenario. And of course, we already have a lot of information that was already measured in different, by different technologies. And so the idea is, okay, we already did a lot of efforts, so why not use it? So, but basically, in all these data sets, for the Cancer Genome Atlas, we have already 10,000 patients. In the NCBI, we have around 1 million samples related to humans, so it's a lot of information that is already there for us to use it. And most of them have an expression profile that is talking about all the picture. So the idea is try to decompose in these integrative cell types. And we can do this computationally. So by using the NCBI, we look for pure samples of the specific cell type of our interest, and we can retrieve the specific expression profile of these cells and build a kind of signature matrix which holds what is expected, a unique profile for each of them. So then we can use this profile in order to deconvolute or to dissect the cells inside the tumor. Basically, it's a very well-known linear model. And the question here, or the unknown part, is what are the proportion of each of these cell types that is in this sample. So there are already a couple of tools to do that, very well known. Timer, CyberSort, and QuantiSeq. These are the most more well known, and this is quite new. So our idea was, okay, we already have these models. We are going to see if they can work for us. So the first thing that you may, or the first question that you may ask to this kind of tools is if they can identify itself, so self-determination. So in order to do that, the model should recognize itself. So instead of the Y, we put each of the columns of this matrix, and they should recognize this coefficient as 1, for instance. So we perform this test, and we see that most of the methods has problems. We expect only this diagonal with numbers and then nothing. But we see that most of the methods, this is timer, for instance, this is a second version of the model. This is linear ordinary least squares approaches. We see that they provide us a lot of more lot more cell types when we expect only one. And basically, the problem is a well-known problem in the prediction literature, which is called collinearity. That this is the autocorrelation matrix of two signatures already available in the literature, which is that with this signature we can estimate 22 different cell types from the immune systems, and with this signature 10 cell types of the immune system. And we see that most of them has a very correlated cell types, expression profile of the cell types, and they severely affect the prediction. QuantiSeq, which seems to be very nice, but also has some correlation problems, and they made mistakes when they estimate in this signature, this signature was developed for this type, for this algorithm. So we can see they have problem for self-determination. So imagine if we apply this to a real patient. Another problem is we perform a kind of simulation in order to see if, so we mix known proportion of the signature in order to see if the algorithm are able to identify correctly the amount of proportion and the amount of cell types present in the mixture. And we see that here we have the expected number of cell types, and we see all the methods tend to overestimate the number of cell types present in the sample. So that means that if your subject has 2 samples, the algorithm will tell you that it has between 3 and 11 and 15 cell types present in the mixture, which is a problem, basically. And also the QuantiSeq method, which was developed for this signature, usually made a strong mistake in the estimation of the number of cell types present in the mixture. So here we have the error in the estimation of the real proportions present in the sample, and we see that all the samples has a strong bias toward high values, high predicted values, or high expected values, which is basically a problem of collinearity, because it overestimates the number of cell types. This will negatively impact in the number of, in the expected proportion of the cell types present in the sample. So we see that none of the methods has a good performance. Many of them provide a high proportion when the expected is none. And this is CyberSource, which works a little bit better, but still have this bias problem. So with this in mind, we try to fix this problem. So we develop a new tool, which is called Mixture, which is a machine learning approach with some concept of data science. And we test them in the same scenarios. And we see that Mixture is very robust independently of the tested signature. So we solve the collinearity problem very well. In addition, we see that it shows almost no bias and very low errors compared to the competing methods. And also is very accurate in order to provide us the right number of cell type present in the sample. So at least using LM22, it gives us in the 75% of the cases, at least 2 more cell types present in the sample compared to the truth. Another, well, sorry for, this is in Spanish. This is a kind of recycle problem of the presentation. So one of the problems of testing algorithms is that in biology we don't have the truth. The gold standard doesn't exist in biology because biology is biology. So when most of the methods that are tested by simulations, but everybody knows that you will perform a simulation that be benefit for you. That is usual. So if you read the paper, you will see that they perform a simulation, and the best method is the one present in the paper, because the simulation is very attached to the model. So, but in this case, we have the opportunity to test, and simulation is always testing positive examples. So we need to test them in the false positive case. And we perform this using the genomics of drug sensitivity in cancer, which are all cell lines. And in a cell line, you don't expect to have any immune cell. So it is our negative or a null distribution test. So we have here around 1,000 transcriptome from different cell lines of different tumors. So we use this to test the different algorithms. And here you have, for instance, the number of estimated cell types for each of the algorithm for this, in this scenario, for this false detection test. And we see that most of the algorithms overestimate significantly the number of cell types. For instance, this is QuantiSeq, and this will tell you that in the cell line, you will find around between 4 and 20 cell types of the immune system for the LM22 signature. And in the case of the TIL10, which you expect at least 10 cell types, you will see that you will reach 10 cell types for in a cell line that you don't expect any. But, and also, for instance, QuantiSeq, in spite they provide a lot of cell types, the proportion are very low, which is good. But it is very dependent on the cell type of the used cell type. And this is the the signature developed specifically for QuantiSeq. And we see that it provides a lot of high values of proportion for these unexisting cell types in the sample. And mixture is one of who provides the less number of cell types. Of course, it's not good, because we expect not to find any cell type, but is the one who provides the less number of false detections, and these detections are very, very low, so we can neglect this estimation. So once we evaluate that our method performs quite well, so now we are using it to explore the immune system in tumor cells, in tumor samples. So one of the things that we are working now is try to characterize the immune system in different cohorts. And here we have, for instance, the number of cell types identified of the immune system identified across all the tumors in the TCGA. Here we have 10,000 samples of 33 tumors. And we see that, for instance, we have glioblastoma and low-grade gliomas, which have more or less the same number of cell types predicted. Kidney tumors, two types of kidney tumors, we also have more or less the same number of cell types. And lung cancer, we also have two kinds of lung cancer, share more or less the same number of cell types. We are still trying to analyze this information. But then we perform an algorithm that allows us Disney is a tool that is used to visualize if we can find groups of in a data set. Here we analyzed all the immune estimation from all the cancers together, and we can see that we don't see anything. All the samples are mixed all around, so the main message is that we don't have an immune content that reflects tissue of origin. And this is quite interesting, because transcriptomic usually talks about the tissue of origin. But the immune system doesn't. So then we start to explore in this cloud the different tissues. And for instance, we found that lymphoneoplasm diffuse large B-cell lymphoma, this is located in this area. This is the profile, the whole profile of the whole subjects in the TCGA. We see that, of course, we find a lot of B cells present in the sample, which is expected because it's a tumor of this, on these cells. And also, we found that we have some macrophages present in the samples. But if we take a look in a closer fashion, we see that we can split these samples in basically 3 groups with very different immune profiles. For instance, we have one of the samples that has very high B cell levels of, I don't remember the name right now, but we have 2 kinds. With this, it's very different from this one and from this one. So this is basically very low value levels of B cells in these samples, but very high values of macrophages M0, which is very different from this one. And now we are trying to explore, this is basically, I put this slide because it's a kind of validation that we are identifying the cell type that we have, that we expect it to be here, that we can see different profiles of the immune system. So we have to explore a little bit more to see what are the effects of this in the phenotype. And we do this in another tumors which are more interesting for us. For instance, glioblastoma multiforme. This is the profile of the whole cohorts. Yeah, basically a lot of macrophages M2, M0, monocytes, and plasma cells, which are basically present in this cell. For instance, we don't have any of B cells. But if we take a look in more details, we can see that we have also different groups with very different immune profiles. And for instance, these profiles provides larger survival to the subjects. And this is very, very interesting for us because we have a platform for analyze glioblastoma tumors that we are right now doing whole exome and methylome analysis. And we are going to incorporate this because we want to select these subjects in order to put them on in a clinical trial. So we are now incorporating this tool into the pipeline of this precision medicine environment for this kind of subjects. Here we have low-grade glioblastoma. So if we take a look deeply in a small number of subjects, we see that they have very different survival profiles. So the immune systems is providing us information about the outcome of the subject, and we are thinking to use it as a kind of biomarker. In fact, the main target of us is try to identify biomarkers or techniques that allow us to follow the subjects or to split them in order to provide them the right therapy. And we can see that, for instance, this group, which is this one, as expected, has a very high values of M2, which is a tumorigenic cell. But this one, which has a little bit more, a better outcome in this part, it has an increased population of plasma cells. And we have to see what happened with these guys, which is very, very bad outcome. So we are still interested to see what happened here. And in addition to that, to this kind of information, we are now able to correlate this information with other biomarkers, for instance, PD-L1 or PD-1 or other markers related to the immune system, and also markers related to chemotherapy. because it's an increased interest in to mix both therapies. One to kill the cell from going to the replication part, and others try to the immune systems kill the cells. Here we have acute myeloid leukemia, that we also have very different profiles in different groups, very well defined. Yeah? And for instance, by means of this method, we can identify a kind of monocytes, high monocyte proportion, which is a new way to identify different types of acute myeloid leukemia. So we can use this tool to identify these kind of subjects. We are also very interested in skin cutaneous melanoma because immunotherapy has nice results in this kind of subject, but only in a small proportion of them. So, and in another proportion, the treatment fails very dramatically. So we are trying to figure it out what is going on there and how they speak the different cells between each other, and what can we do to improve the treatment. For instance, right now there are some drugs that can switch the behavior of the macrophages in tumorigenic and anti-tumorigenic way. And we see, for instance, that these two groups have very different survival profiles, and the worst one has an increased amount of macrophages M0. And this one, which has a better profile, has more proportion of CD8, T, follicular helper, and TREX cell types present in the sample. So this can be used to guide the therapy. This is our expectation. In lung adenocarcinoma, also, we have two populations with different profiles. In the survival, but also in the behavior of the immune system, with an increase in plasma cells for the case of these subjects, and here the macrophage M0, which is worse at the beginning, but then seems to reach a plateau. So these are things that now we are trying to relate to other biomarkers in order to build a kind of a guide or something that allow us to figure it out. What is going on in these scenarios? We also found that the level of macrophages M0 is related to high number of variants in the samples. So it is known that more number of variants also is related to some kind of messages between the immune systems and the tumor. So it seems that everything is related to everything. So we are trying to figure it out that too. So this is a kind of forecasting that we're going to see then. But we are also evaluating the immunotherapy response in melanoma, and we were able to identify that those subjects who respond to the therapy, these are the subjects in the previous to the therapy, and this is on therapy, and we see that the responders has on treatment has a lower level of macrophages M2 compared to the non-responders, and we also find that CD8 increase in the responders against the non-responders. Gamma delta also increase in responders compared to non-responders. So this is our very recent results last month. So we are very happy with the tools. I think we can. It's going to be very useful in the near future. So we have a lot of work to do because our idea is to try to figure it out if we can characterize the immunosome of tumor samples in order to get more knowledge about them and to see if we can move toward some kind of therapy guidance. Mixture is available for everybody. You can download from here. is a Python application, but also we have an R application. It's very easy to use. You just have to prepare your samples in an Excel file, all the transcriptome, RNA-seq, or macroarray data. I suggest from Affymetric platform or from one-color platforms. We cannot evaluate yet in Agilent technology, but I think that it's working as expected. So then you just drag the files and set the parameters, and then we'll give you a lot of information that you can download and process. You have the proportion of the cell types present in the sample, explore how they distribute in each sample. Well, many different type of plots. We are not going to stop to dig in this. So it's very short, the talk. I want to thank my collaborators, Dr. Girotti and Jamil, which is there. So I'm happy that he can come here to share all the experience with you. This is the lab at the CDA, and these all the financial agencies, and well, other groups, other partners in a national university. So, well, this is all that I was thinking to share with you. Thank you very much for the invitation, and we hope we can see us again in the near future.
Thank you very much, Elmer. Some question from the floor? Yeah. Question here.
Thank you, very nice talk. I was curious for the research on low-grade glioma, because I was working on myself on that as well, but I'm not a biologist, so maybe it's a stupid question. But you showed that for your immune markers, they influence the prognosis. And from what I understood, it's also well known that the certain mutations, like the IDH mutation and the 1p19q mutation, also influence the prognosis. So I was just wondering if are those two related? So is the 1p19q mutation also related to this immune marker, or is that a completely independent marker?
Well, it's a nice question. I don't know. Okay. This is a result from 2 weeks ago. Okay. So, but what I know that Jamil already looked for, not for low-grade glioma, but also in another tumor, that we have a relationship between the markers, mutation markers, and the immune system. And in breast cancer, for instance, we saw that if we use first a gene, the state of the gene mutation or whatever, to split the population first, and then use the immune system to re-split this population, we have a very good identification of patients who might benefit from this particular therapy.
Okay, yeah, thanks. Very interesting. Maybe we should talk later. Yeah.
So you can use it and test it.
Thank you. It was very nice. I got interested in to know, in the same patient, you see higher CD8s and lower M2. Is that a different significance of? Because we saw the different in different pictures, but I mean, if you combine the three information, how maybe the numbers
you mean to relate that?
Yeah, I mean,
the proportion of one cell type in the same subject against the other.
Yes.
Well, we don't have a conclusion yet on that. There are some papers that use this kind of relationship. But this paper are using the other technology, so which is biased. So up to now, we don't see very strong patterns relating to different cell types. For instance, in the case of macrophages, I mean the relationship between M0 and M2. We have a suspicion that M0 is not really M0. something in between. It's more like M2 than M0. We can prove that, but apparently is what we are seeing right now. So we are working on that to see, because our intention is to identify some kind of biomarker or something like that.
Other question? Yeah.
Nice too, Elmer. So how easy it is it customizable enough for you to provide signatures, for example, of exhausted CD8 T cells and change the whole stuff to quantify specific subsets within the tumor microenvironments?
You can use this algorithm with any kind of molecular signature if they pass all the tests that I mentioned before. It could be a good partner with the algorithm. Related to measure the exhaustion, these signatures doesn't measure exhaustion, so we are using, for instance, some other biomarkers to evaluate them, but we still don't have any conclusive finding on that. But exhaustion is a very hot topic, but there is still no model or algorithm or signature that give us good information about it, but everybody knows that it's something that should be there. Something that we are using to see, for instance, if we can stimulate the PD-1 environment is to monitor, for instance, there are two OX40, and its ligand. And we found some relationships that tell us that we can, for instance, stimulate PD-1 or PD-L1.
Other questions? So I have one. I have a question. So I think your case is interesting because it shows that instead of just having the stratification of the patient based on the transcriptomic, you use the transcriptomic to define what are the immune cells present in the tumor microenvironment, and then based on this cellular analysis, you stratify the patient. So it is an additional step in the stratification process. I have a question. Do you have example where, based on the understanding of the presence or absence of a given immune cell type, it's possible to drive the treatment option, or it's too early to have such example? For instance, in glioblastoma, there is one, I don't remember which was the cell type present. Is it possible to try to handle this, to impact the number of cells in the tumor microenvironment by immunomodulator or cytokines and so on, and to reinforce the treatment?
We don't have any experience on that, but this is something that is already under research in worldwide. There are several techniques that intends to stimulate the immune system or to switch the M2 to M1, which is more anti-tumorigenic. So many people are working trying to modulate the immune system of the tumor. I think this kind of tools is going to give you an idea of what they have to do, and probably in the near future we can go further in this area. Yeah.
Okay. Thank you. Anyway, other questions? Okay. Thank you very much.